Well, welcome back. This is Chris Wozniak again. Now we're going to turn to a little more specific coverage and discussion of the registration process for plant-incorporated protectants at EPA. First, Mike Mendelson will give an overview of this registration process, and then Elizabeth Milewski will follow up with some details on new efficiency measures for that registration process. So, Mike, please take it away.
Thanks, Chris. Okay, we're going to talk about the registration process for PIPs. And PIPs, as we mentioned prior to this, are pesticides and fit into the normal regulatory scheme at EPA. At the bottom of this page, you'll see a link to our registration manual. There's a lot of good information in there about PIPs and about the registration process. And this page basically gives an overview of what I'll be discussing. And as part of this, there's some pathways to make it a little bit easier for smaller developers, fee waivers, and other things. So we've tried to tailor this presentation to provide some strategic guidance in registering and getting field approval for PIPs. So we'd like to talk about commercial development in EPA, registration under FIFRA, pre-submission meetings as we discussed previously, various types of PIP products, the actual package you'll be putting together, the petition package. Again, there are two statutes or laws that we operate under primarily, and there's two different packages that are necessary. They come in together, but you're having to address both the residues in food and also the licensing part. We'll talk about experimental use permits and field trials, fees, our process with completeness reviews and screens, data formatting requirements, confidential business information, transparency, and then new efficiency measures. We'll talk particularly about the IR4 assistance program and what's available, and then take questions and answers. So again, as mentioned previously, EPA's oversight focuses with PIPs on the pesticidal substance, such as the Bt protein, and the genetic material necessary for its production, such as the CRY gene. This slide demonstrates commercial development in EPA. So as was mentioned earlier, during your product design phase, we encourage you to consult with EPA during your research and development. As we move down the process, there's experimental use field trials, and when you have field trials and residues may be in food that may be distributed, there's a temporary tolerance exemption requirement. So even though the product is not completely registered, it's under field trials, we have to address the residue in food, and there's a temporary tolerance exemption process for that. Moving along the line here, there's step 3 would be seed increase or breeding registrations and tolerance exemptions. So with PIPs, unlike conventional pesticides that can be produced in brick and mortar facilities, PIPs are produced in the field as far as the seed increase and growing those up, and those are regulated as registrations. And finally, the last step would be a full registration for commercial use by farmers and growers and a tolerance exemption. So again, the different statutes we operate under: FIFRA, the Federal Insecticide, Fungicide, and Rodenticide Act, also the Federal Food, Drug, and Cosmetic Act. Again, FIFRA focuses on the licensing or the registration or the experimental permit for field tests, and FFDCA focuses on the residue in food. Section 3 of FIFRA is the portion of the law that deals with registration. Section 5 deals with experimental use permits, and Section 33 deals with the fees and the timing of the applications, which was the amendments to FIFRA under the Pesticide Registration Improvement Act, or PREA. Under the Federal Food, Drug, and Cosmetic Act, the tolerances or maximum residue levels or tolerance exemptions for pesticide chemical residues in or on food and feed is under Section 408 of the Federal Food, Drug, and Cosmetic Act. So next I'd like to talk about PIP registration and requirements. If a PIP is tested in a field at greater than 10 acres, an experimental use permit is required. If a PIP is distributed or sold for commercial use, including seed increase, a pesticide registration is required. And if a PIP is used in a food or feed crop, or tolerance exemption from the Federal Food and Drug and Cosmetic Act under Section 408 is required. So as was mentioned earlier, coming in and talking to us early is something we highly encourage. At EPA, we consider that a pre-submission meeting. Determine what data is necessary to support EPA's risk assessment. So the focus that EPA has is that in order to allow for a field test, or to allow for commercial use, or to allow for the residue of the PIP in the food to be consumed by the public, EPA conducts a risk assessment looking at hazard and exposure. These pre-submission meetings are confidential, and at these meetings a determination of data needed to evaluate whether a PIP can meet the FIFRA standard and the FFDCA standard is undertaken. These are very helpful early in the process. The types of PIPs we've registered today are varied. Different crops such as corn, cotton, potato, plum, papaya. We've also, in these crops, had single traits, multiple PIP traits affecting different pests, which we call stacked, multiple PIP traits affecting the same pests or pyramids, which is a very powerful tool in resistance management. We've had seed blend products where non-PIP corn is mixed with PIP corn, or different types of PIP corn seeds are mixed together in order to accomplish insect resistance management objectives. We've had seed increase or breeding use registrations where the use of the product is to grow up seed to produce hybrid seed later for farmers. We've had commercial use registrations which allow for the PIP plants to be sold to farmers or to nurserymen. And also, as we look at PIPs in the registration process, is this concept of the old active ingredient and the new active ingredient. Sometimes there are different variances of a particular PIP active ingredient, and so in those cases, the registration requirements are not as difficult. Again, any pesticide product with a new or not currently registered active ingredient generally requires a more extensive review than products containing already registered old active ingredients. PIP products may contain new active ingredients, old active ingredients, or both. So now I'd like to talk a little bit about the registration process. The seed increase registration enables seed companies to produce enough seed for distribution to farmers. If the pesticide is contained in a food crop, a tolerance or tolerance exemption must be in place. For insect resistance management purposes, trends towards combined products, many different traits in a particular PIP. And also we'll be talking later today about a new registration process for PIPs and breeding line intermediates. That's been put out for comment. EPA proposed an alternate approach for regulating these breeding line intermediates that involve individual trait product registrations used to make the BLI and eventually the commercially stacked or pyramided hybrid. So now I'd like to focus on how you put an application package together. The administrative portion of the registration application package, kind of a nuts and bolts approach here. With an application, and again, one of your greatest resources here is the Pesticide Registration Manual, which you can find on the URL on the first page of this presentation. But the package is put to several components. One is the fee payment, again under PREA. For pesticide registration actions, there's a fee that's charged. There are some waivers of these fees and reductions we'll talk about later. These fees are required for EPA to review the application, and also associated with those fees are specific time frames for EPA to make a decision about the application. A cover letter is necessary, an application form, and there's a URL here if you just in this presentation to get there, the application form for the pesticide registration. These are all components that are necessary in the registration application package. Draft labeling is required. The data or literature that's being used to support the particular application. Another form that we call a confidential statement of formula is necessary. For product registrations, applications, a certification with respect to citation of data form. This form has to do with how you cite data. If you're not generating the data yourself, if you're maybe citing other data that's been submitted by another company. The data matrix that lists out how you address the various data elements in your application. And then data waiver requests are part of the package as well. So in addition to a registration package, when applying to commercialize a PIP, one needs to file a tolerance petition package if the PIP is in a food crop. There is no application form for petitions, and requests must contain the information listed in 40 CFR 180.7. Please refer to PR Notice 97-1 for information on filing a tolerance petition under the Federal Food, Drug, and Cosmetic Act. This act requires that the petitioner submit an informative summary of the petition and the data and any other information and arguments submitted or cited in support of the petition. Essentially, when you submit a petition package, you put forward a risk assessment for EPA to consider. That's a risk assessment summary. The next portion of this slide has a company notice of filing petition summary template. So somebody who is putting together a tolerance petition package would then fill out this template, and in addition to the cover letter and all the other registration information, there would be a company notice of filing petition that would be submitted. And this actually is put into the docket when EPA announces its notice of filing for the pesticide petition to allow for residues in food. All right, next I'd like to talk about when an experimental use permit is required. For PIPs, it's required when there's testing on a cumulative acre total of over 10 acres for pests that occur in different geographic locations over 10 acres per pest. So what that means is if you have a different sort of pest in California than in Michigan, that you could have, you would have to be over 20 acres in order to trigger an EUP requirement if you're testing 10 acres per site. So again, that's a little nuance there, that for pests that occur in different geographic situations over 10 acres per pest. A tolerance or tolerance exemption may be needed when an EUP is not required if the PIP could enter the food supply. So it's important that the PIP, when being tested at small scale, not enter the food supply either through distribution of the seed or plant material or through the flow of pollen to other adjacent crops. EPA feels that when these small-scale tests are done under USDA authorization, that the containment is adequate. So again, just mentioning about field testing containment of pips and pip pollen. If you don't have a tolerance exemption in place for the particular pip, outcrossing of pip pollen must be prevented regardless of the test plot size. For corn, spatial isolation, reproductive isolation, and temporal isolation are used. Without a tolerance or exemption, harvested crop must be not enter commerce. Now, with EUPs, which are over 10 acres, these typically have temporary tolerance exemptions in place with them. It's very rare now for a EUP for a PIP not to have a temporary tolerance in place. At the bottom of this page is a link to a pesticide registration notice. It's 2007-2, which provides guidance on small-scale field testing and low-level presence in food. Again, the concern here is that at small-scale testing, that the PIP does not enter the food supply without an appropriate tolerance exemption. Experimental use permits enable developers to generate data for full registration. EUP data usually is a subset of the registration data, including information reviewed for the tolerance exemption. It may be presumed that an EUP is not required at or under 10 acres of land or 1 acre of water. Again, EPA evaluates all active ingredients. Inerts for PIPs are frequently herbicide-tolerant selectable markers. Now I'd like to talk a little bit about the experimental use permit application. So with this application, there's, again, a fee payment under PREA, a cover letter, an application form for the experimental use permit, draft labeling, data, another confidential statement of formula, the data matrix. In addition to those components, an EUP requires a Section G, and the Section G is the description of the experimental program, as well as data waivers. But again, the key, one of the key components of the EUP application is this Section G, which really outlines how the experiment is going to be performed and who is going to perform it. In the Section G, or that's the Section G of the application, but we call that the experimental program, participants and cooperators are listed, names of states in which the product will be used along with the amount of active ingredients and acreage, the names of the states in which the pesticide may be shipped for further distribution. So this EUP authorizes the shipment and the use of a pesticide for experimental purposes. It has information on prior testing, objectives of the experimental program, should provide justification for the quantity of active ingredient proposed to be used under the EUP. Again, the EUP is to provide data to support a registration. How many acres is being required under the EUP needs to be justified under Section 5 of FIFRA, which outlines the requirements for an experimental use permit. The Section G, or experimental program, will also provide details concerning the method of field trial confinement and disposing of unused materials at the conclusion of the testing program. For instance, checking for volunteers after harvest, isolation distances, those sorts of things. At the bottom of this page, we have a link to what we call Tips for Plant Incorporated Experimental Use Permit Program Submission. This is on our website and provides additional guidance as far as how to calculate the acreage and other information. Again, PREA is involved with fees, and here is a link for the Pesticide Registration Improvement Act, coupled the collection of individual fees with specific decision review periods, and these provide links to those fees for registrations and experimental use permits. All right, this next page provides examples of PREA timeframe and fees for PIPs. So, for instance, for what we call a B771, that's our PREA code. That's an experimental use application for a new PIP with a petition to establish a temporary tolerance. That is charged $127,000 and $630. A new PIP for non-food use is about $160,000. A registration application for a new PIP with a petition is about $320,000. And you can see here some various other examples of PIPs. An application just to amend a registration, to change it, including extending the expiration date or modifying a plan, about $13,000. So these are all different fees that are required for the various regulatory actions. And the previous URLs provide a more detailed list of the various types of actions that we see. Well, as we talked about the various fees, now we'd like to talk a little bit about fee waivers and refunds. So there are different avenues that one can pursue to reduce or eliminate the fees under PREA. A reduction of 50 to 75% in the fee can be granted for a qualified small business. And again, the registration manual provides links in the fee section. Applications from federal and state agencies and those solely associated with the tolerance petition submitted by IR4, where the fee exemption is found to be in the public interest, can be exempted from PREA fees. So federal and state agencies and those that are submitted with IR4 can be exempted from PREA fees. Fees may be reduced or exempted for minor uses. An EPA has a discretionary authority to issue a partial refund up to 75% of a PREA fee on the basis that in reviewing the application, the administrator has considered data submitted in support of another pesticide registration application or submitted in support of an application pending with the agency. All discretionary refund requests must be requested up front and be included in the application. So this is an important point. If you're relying on other data that's been submitted with other applications and you feel like EPA has already evaluated that information, you can request a partial refund. But that has to happen up front at the time you make your application. In addition to PREA fees, FIFRA has maintenance fees. So the PREA fee is the fee to review the particular pesticide action before the agency, a registration or an experimental use permit, or perhaps changing or amending an existing registration. Maintenance fees are in place for every pesticide product that's registered. All Section 3 and Section 24C, 24C pertains to state registrations, are subject to an annual maintenance fee. The fee for 2016 is about $3,500 per registration. So that's charged and billed by the agency on an annual basis. Maintenance fees will be reduced by 25% for the first registration only for qualified small businesses. Maintenance fees will be reduced or waived for minor agricultural registrations. When EPA determines that the fee would be likely to cause significant impact on the availability of the pesticide for the use. Maintenance fees for certain public health pesticides will be waived if based on information supplied by the registrant that the economic return to the registrant from the sale does not support the registration or re-registration of the pesticide. For further information, see the hyperlink below here on maintenance fees and ways to have reductions or waivers. The next thing I'd like to talk about is the process. So once you've submitted your application, there's a very important process that's been implemented under PREA, our completeness reviews. We have, and several of these completeness reviews are kind of critical in our process. The first is the 21-day content screen, where we determine whether the appropriate fee has been paid and that the application contains the necessary forms, draft labeling, and data formatted in accordance with the agency guidance. Applicants must correct applications within the 21-day initial content review period to successfully complete the screen. And it's critical that you successfully complete these screens to move through our registration process. Next, I'd like to talk about the completeness screen, or the preliminary technical screen. After the 21-day content screen, a preliminary technical screen is conducted for PIP applications within 90 days after the 21-day content screen is completed. These determine if the application and accompanying information are accurate and complete, consistent with the proposed labeling and a tolerance or intolerance exemption, and such that the product to full review could result in the granting of the application. This technical screen is a critical juncture in your application. If the application fails the technical screen and the deficiencies cannot be corrected by the applicant within 10 business days after receiving the agency's notification of deficiencies, the agency will reject the application. This provision of PREA was put in place to encourage complete submissions being reviewed to the agency. So it's important with a PIP application not to submit a partial application, to get your foot in the door, to have the application here. If the application is not complete, when EPA does the completeness screen, if it fails that screen, the application will be rejected, and you will lose a portion of your fees and have to start over again. So it's really critical in the process that the application, that the package be complete, that you go through the pre-submission meeting, and you address all the relevant data needs to support the particular PIP or experimental use permit application. Next, I'd like to talk about data compensation, another aspect of the registration application. Exclusive use and compensation rights and data submitted to EPA to support registration and experimental use permit actions. Now, data compensation is not required to cite data supporting an experimental use permit, but data submitted with an experimental use permit application is compensable if cited in support of a registration application. Exclusive use protections mainly apply to those data that are submitted to support the initial registration of a new active ingredient. Compensation rights extend to data submitted to support or maintain a registration or an experimental use permit. Lastly, the Food Quality Protection Act of 1996, which amended FIFRA and FFDCA, amended the data protection rights of FIFRA by expanding exclusive use rights for minor uses. Exclusive data compensation is a way to compensate those that have generated data to support a registration or an experimental use permit. And if you did not generate and submit the data yourself and you're citing it, typically if the data has been submitted within 15 years, there's an obligation to pay compensation. Next, I'd like to talk about data formatting. Many may think that formatting the data is an annoying administrative part of the application, but if it's not done correctly, it can significantly delay the review of the submission. Pesticide Registration Notice 11-3, which you can access by the hyperlink here, contains specific instructions on organizing and formatting submittals of supporting data. And I want to mention here also, all these presentations are currently present in the docket that is listed associated with this webinar and also the September presentation on the PIP data. Submission of data to EPA is made with the understanding that the performing laboratory may be inspected to ensure compliance with good laboratory practices. There's a good laboratory practices statement. It's a very critical statement. To be truthful with that, there are penalties if you're not. Common errors with data formatting: incorrectly formatting confidential business information statement, missing or incomplete GLP standard compliance statements, incorrect pagination, legibility problems, unsigned documents. I mentioned about the first one here, incorrectly formatted confidential business information statement. Sometimes what happens is the confidential business information statement says there's no CBI, no confidential business information in the document, and other places in the document, the pages are marked confidential. It's important to follow this PR notice and have the formatting done correctly. Now I'd like to talk a little bit about confidential business information. Section 10 of FIFRA provides applicants with the opportunity to claim information as confidential and requires EPA to protect information. Section 10D1 also provides that safety and efficacy information on registered or previously registered pesticides must be available to the public. The exceptions to this mandatory disclosure are information on the manufacturing or quality control processes, methods for testing, detecting, and measuring the quantity of deliberately added inert ingredients, and the identity or percentage quantity of deliberately added inert ingredients. In addition to confidential business information, FIFRA has a Section 10G, which prohibits the disclosure of certain information submitted by an applicant or registrant to any employee or agent of a multinational pesticide producer, seller, or distributor, or to anybody who intends to deliver such information to a multinational pesticide producer, seller, or distributor. What does this mean? This means that even if the data that's submitted is not confidential, EPA cannot release the data to one of these entities that cannot, that's related to a multinational pesticide producer. So again, EPA protects the confidentiality of confidential information, but also the data. And the reason for that is Congress put this protection in the law so that somebody could not take the data that was been generated by another party and then submit it to perhaps a foreign regulatory body to obtain approval there based on the data. So it's kind of a patent-like protection for pesticide safety data. Now, for PIPs, there's a special provision for confidential business information under Section 40 CFR, the Code of Federal Regulations, 174.9, that any claim of confidentiality must accompany the information at the time the information is submitted to EPA. Failure to assert a claim at that time constitutes a waiver of confidentiality for the information submitted, and the information may be available to the public subject to 10G of FIFRA. So the claim of confidentiality must be made at the time of the application. If you don't make the claim, we don't consider it confidential and can release it to the public. Any claim of confidentiality must be accompanied at the time the claim is made by comments substantiating the claim and explaining why the submitter believes that the information should not be disclosed. The submitter must address each of the points in 40 CFR .204E4 in the substantiation. EPA will consider incomplete all PIP submissions containing information claimed to CBI that are not accompanied by substantiation. So having the substantiation is critical if you're making confidential claims for a PIP application. If you don't have the substantiation, EPA will consider the application incomplete. And if this is not rectified either during the 21-day context screen or during the 90-day technical screen, the application will be rejected. The next slide talks about the various components that the submitter must address each of the points in the substantiation. And it goes through the various points that have to be addressed to substantiate why the information is confidential. Lastly, I'd like to talk about transparency. In the PIP registration process, the full risk and benefit assessments for new active ingredients are published on our website, as well as records of the scientific advisory panel meetings on these products. The next bullet has a URL for the OPP public participation process, and we continually publish notices in the Federal Register consistent with FIFRA and FFDCA. For example, registration applications and decisions, tolerance exemptions, notices of filing, final rules, EUPs, etc. Now I'd like to turn it over to Elizabeth Maluski, who will give a talk on efficiency measures under consideration by EPA.
Okay, thank you, Mike. So I'd like to talk to you a little bit today about efficiency measures that we're currently considering. As the technology has developed and we've gained more experience in regulating PIPs, it occurred to us there may be some areas where we may be able to increase efficiency, both for the agency and for registrants. And there are two areas that we're looking at primarily. The first one is the way that we view transformation events, and the second is the way that we look at intermediates for breeding stacked or pyramided products. And we call those intermediates breeding line intermediates. Why am I having a problem here with this? Mike, why am I having a problem with this? Thank you. Okay, so there are some considerations in the transformation event that I'd like to talk to you about. Currently, any change in the 5 pieces of information that are listed there generally means that we treat the PIP as a new active ingredient, and as Mike has discussed with us, that has implications for costs and for the amount of time involved in the process. So those 5 pieces of information that we generally consider now in identifying a PIP are the active ingredient, any inert ingredient, the transformation event, the vector, if any, used to affect the transformation, and the plant. But recent advances in sequencing and modification techniques offer options on how we might view the transformation event and the other information that is required. So, advances in sequencing information in particular allow us to consider information showing that a PIP from a different transformation event is the same active ingredient as an already registered PIP. If the PIP is identical to or substantially similar to an already registered PIP, we can determine that by looking at submitted sequencing information, and we can then categorize it in the different PRIA categories in ways that may be more simple in terms of submission. For example, if we find that it's the same active ingredient as an already registered PIP, we can register that particular PIP as a new product. And therefore, it comes into a different PRIA category with different costs and different timelines for evaluation. It doesn't like me. I don't understand. Thank you. Sorry, folks. Technical difficulty here. All right. So in talking about the transformation event, here on the slide, we can see that there is the gene of interest, and that particular gene of interest is carried on vector X. And the developer wants to introduce it into 3 different potato varieties, 1, 2, and 3. Under the current system, that would mean a new product registration for each of those potato varieties because it's a different transformation event. However, under this new approach, we're thinking that this might be 3 products, but it might be simply 1 active ingredient. So basically, multiple transformation events under 1 PIP would equal a simple 1 active ingredient. So again, as I've said, implications for the PRIA category that it's assigned to, and there are implications for exclusive use and data compensation. Now, this approach can apply to PIPs in both seeds and clonally propagated plants, and it's particularly useful for the clonally propagated plants, as you can see above in our example for potato. It would mean that it would be 3 products but 1 active ingredient rather than 3 separate active ingredient registrations. Under the approach, each of the identical or substantially similar PIPs, for example, each new product, would still be associated with its unique transformation event identifier. Okay, try again. Mike, you're going to have to help me again. I don't know why it does not like me. Okay, so when we look at the transformation event and think about how we might use it in this more flexible manner, the determination would be made by EPA on a case-by-case basis. It would be based upon data that the applicant supplies to EPA showing the equivalence of the PIP to an already registered PIP. In other words, it's either identical or substantially similar to an already registered PIP. The data would have to be submitted comprehensive molecular sequence analysis using high precision techniques. And the developer would have to show there are no anomalies that might affect the inserted genetic information. For example, there should be no open reading frames. Also, tissue expression profile has to be the same, and the expression levels within the plant in the various tissues have to fall within the same certified limits. The modification also has to occur within the same plant. In other words, the same species. So information on this more flexible approach will hopefully be included on the website with this webinar in the near future. It's going to be a white paper that describes some of this in greater detail. So now to move on to the breeding line intermediates. So on this slide, we show one of the issues that arise with breeding line intermediates. As you can see from the graphic, if you have 5 different PIPs that you want to stack together or pyramid together into a single final PIP product, you can go through various breeding line intermediate stages in order to bring those PIPs together. And as you can see, the number of potential combinations is quite large. Currently, we require a unique registration for each single trait PIP and for each combination of 2 or more PIPs stacked or combined together. So theoretically, there could be quite a large number of breeding line intermediates that could be involved in the creation of a single final product, and that means quite a few different registrations for those particular breeding line intermediates. So this is also one of the areas in which we are looking to see if there are ways that we might make the approach more efficient. I have no idea why it doesn't like me, but it doesn't. Thank you. Okay, so when we consider this new approach, basically a new registration, a unique registration, would no longer be required for each combination of PIPs in BLIs. Rather, PIPs and BLIs would be regulated to the registration of the individual PIPs to be combined to make the commercial stacked pyramided product. Each individual PIP that is part of this breeding strategy, so those that are on the left-hand side of the graphic, would have its own unique seed increase registration. Each of those single registrations would list the other PIPs with which it can be combined. A separate registration, a commercial registration, would still be needed for the final stacked PIP end-use product of the breeding. Okay, so I've got some information on this slide that shows where you can actually find a white paper describing in detail this particular efficiency approach. It gives the docket number. I'm sorry, but the comment period is now closed. We have received a number of comments, many useful comments, and we're in the process of considering those comments to try to determine the future approach and to see if any modifications are needed to what we suggested. So basically, that ends my presentation. Thank you.
Do we have any questions from any of the attendees on the registration process? Oh, you're going to do that. All right. Actually, Mike Mendelson's going to give us a little presentation regarding IR4 as part of this process.
Thanks, Chris. Yeah, so we want to talk a little bit about, I mentioned earlier about the IR4 process and the what it can do. So this is a presentation that was sent to us by Dr. Michael Braverman with the IR4 project. And we just wanted to highlight some aspects that the IR4 programs and benefits it provides for PIP registrants. As mentioned here, there are 2 significant hurdles for public sector, especially crop biotechnology. The expertise needed to prepare a package and the PREA fees. So IR4 has helped with both, and it's a resource that's available to folks. Again, this is information that you can look into at another time, but IR4 has assisted in the registration of the plum pox, honey sweet plum, papaya ring spot resistant papaya. Also, when IR4 submits a petition to EPA associated with the registration package, and it's in the public interest, the submissions are exempt from PREA fees. IR4 does provide regulatory assistance in the case of public interest without charging consulting fees. So I think it's important to realize this is a resource particularly for government and academic institutions that with minor crops, that it's not, IR4 does provide support with the putting the package together. And the advantage of submitting a package with an IR4 petition is that you don't pay a PREA fee. IR4 requires a public sector request, a letter explaining the need, why the registration is in the public interest, and preliminary efficacy data. The request is then reviewed by the IR4 Project Management Committee and grower representatives, and the process is started online. Again, this is not something that EPA is directly involved with. We wanted to present this and have this information available to PIP registrants as far as support in preparing an application, as well as a way to waive the PREA fee. This is an example on the IR4 website as far as how to fill out the form and instructions to do that. Okay, lastly, as part of this session, there's a number of resources we've included on this page that you go back to and look at some of the source documents that were used to produce this presentation. Now I'll turn it back over to Chris.
All right, well, let's pause again here and see, are there any questions from the attendees on any of the registration process or IR4 that we just presented?
In order to ask a question, again, you would fill in the question and answer pod at the bottom there and hit the comment button.
All right, well, not seeing any questions coming forward, I'd like to again thank Mike and Elizabeth for their presentations, and now we're going to hear from Dr. John Cow, Senior Scientist in the Biopesticides Group, talk about product characterization of plant-incorporated protectants.
Hello, everyone, and thank you, Chris, for the introduction. I'm John Cow. I'm going to be speaking to you today about the registration. No, I'm not going to be doing that. I'm going to be speaking to you about product characterization and human health for the information that we use to generate a registration decision document. The product characterization data that I'm going to be speaking to you about is similar to the information that's you've heard about as is provided to USDA and FDA. This kind of information is critical to understanding the way in which a product was made and any unique characteristics that need to be assessed for the registration decision. Information on specific transformation systems and DNA sequence inserted into the plant, inheritance and stability of the traits in the plants, and the biochemical characteristics of the PIP protein, as well as expression levels for various plant tissues, are all items that we consider important for characterizing the product. And all this information provides a basis for possible hazard questions, as well as addressing potential exposure of the PIP through the plant expression process. In terms of the data reviews that are associated with the submission of data, there are a number of different classifications that are provided when the data is reviewed by a science reviewer at the agency. The first is acceptable, and that is basically a sign that the study has been done in a scientifically valid manner and that it provides information that's useful for the risk assessment because of that. Supplemental is a level below that. Basically, it's saying that the study provides some useful data that is going to be useful in the risk assessment process. And the supplemental upgradable says that while there is information that was provided, the study really doesn't provide enough information to fulfill the EPA guideline requirements, and that certain aspects may not be scientifically acceptable as presented. In the case of a rating of supplementary upgradable, there is going to be an indication of what needs to be provided in order to make that study useful for a risk assessment process. There's also, of course, an unacceptable classification, but hopefully the process that was described previously about screening the data for acceptability and usefulness for a risk assessment process will have indicated that status early on in the process so that any kind of information or new studies that need to be provided can be informed to the registrant early on in the process. In terms of product characterization, there are 6 categories that I'm going to be speaking to you today with regard to product characterization. This includes the origin and nature of the trait that was introduced, the method used to introduce that trait into the plant, known as the transformation system, characteristics of the inserted DNA, inheritance and stability of that trait after transformation and regeneration of the plant itself, protein characterization of that trait, and expression values for the different tissues. And finally, residue analytical method, which is used for enforcement purposes. The first category under protein or product characterization is the origin and nature of the PIP trait. The original source of the PIP is very important in terms of identifying any hazards that may be associated with that source itself. Expression elements and marker genes relate to changes that may have been made to the sequence in order to promote effective expression of the PIP trait in the plant, as well as any issues that may be related to marker genes, which are, in the case of PIPs, markers, typically antibiotic or herbicide-resistant markers that are used in the selection process after transformation. Essential to the description of the nature of the PIP is a discussion of the mode of action for the PIP trait. We've seen traits related to expression of coat proteins from plant viruses that relate to the suppression of viral infection. We've seen cryoproteins expressed that are basically pore-forming proteins in the insect gut. And we've also seen disease resistance traits expressed in PIP plants. The next category is the spectrum of activity for the PIP trait. Unlike the mode of action, this is basically the registrant providing information as to what the level of protection for that PIP trait would be in use in the field. So this is basically saying what type of pest species are known to be controlled by the PIP trait and any other information that may be relevant and useful, especially with regards to the non-target organisms that may be exposed. Finally, there's a discussion of the biology of the host plant that has been transformed with the PIP trait, and this is used to basically inform the possible exposure scenarios both in the diet and for environmental non-target exposure issues. The next area with regard to product characterization is a description of the transformation system that was used to alter the plant. This includes a complete description of the PIP expression cassette elements, both the coding sequence and any changes to the promoter or terminator sequence, as well as marker genes and other aids to the transformation process. Typically included is a discussion of a plasmid or other source of the PIP DNA trait and a thorough map of the expression cassette elements, and typically that includes REN or restriction endonuclease activity sites, so that subsequent data that's presented about the characterization of the DNA in the plant can be interpreted. The transformation method specifically used and the selection process after that transformation are also a part of this description. And the transformation method would be something similar to, is it agrobacterium transformation method used, or is it a simple biolistics method in order to introduce the trait? And finally, as part of the transformation is basically the regeneration of the plant and backcrossing for stability and assurance that there are the agronomic traits that are desired. And this is really an important part of the process. It's well known that a lot of plants have been recalcitrant to transformation because it's difficult to regenerate the plant from a tissue culture scenario, so that you can have a successful transformation and then not be able to regenerate the plant. The next part of product characterization, once you've transformed the plant and successfully regenerated the plant from the callus tissue, it's important to characterize what was actually inserted into the PIP expressing plant. And we know that you provided the intended PIP trait and the map with the expression cassette and the REN sites. The next part of this information is actually generating information to show what was incorporated into the plant itself. And sometimes this differs from what the ideal intended PIP insert is. So basically there can be a few deletions, there may be multiple insertions or things like that. It's just a matter of characterizing the actual DNA that's present in the plant. This would also encompass a consideration of copy numbers that were of the insert that were found to be in the plant, any information related to the location with the intent of possibly identifying genes that may have been disrupted, and finally a consideration of any possible open reading frames that may have been created with the insertion process. The creation of open reading frames is important because if it's found that there are new proteins other than the ones that were intended to be introduced being produced in the PIP plant, the safety assessment would have to encompass the safety of those proteins also. For product characterization,
the leader has disconnected. The conference will be terminated in 5 minutes.
Well, I hope I can speed up enough to actually cover that 5 minutes, but we'll see what the snafu is about the leader terminating the conference. Hopefully you can still hear me at this point. Well, unless I hear otherwise, I am going...
I would go ahead. I think we're still connected for 5 minutes at least.
Okay. In terms of the inheritance and stability, we're looking at demonstrating that the PIP trait that has been introduced is predictably inherited according to Mendelian laws in successive generations. And this would also include, would be important for plants that are not sexually reproducing but vegetatively reproducing, and basically showing that over subsequent reproductive cycles for the vegetatively propagated plants that you have predictable expression of the PIP trait. We'd also be looking to see that the genetic material that has been transformed into the plant is stably inherited over time, and that the copy number and location and possible reading frames that have been created remain stable over time. And basically it's showing that the insert that you initially described in the characterization of the inserted DNA continues to produce as expected and doesn't show any untoward expression or loss of expression in the case of something that may have multiple copy numbers of inserts. I'm going to go to the next one, which is the product characterization and expression of the intended PIP trait. Basically, with this data endpoint, we're looking to verify that the intended phenotype or activity of the PIP trait is being expressed. We're also looking for quantitative expression levels in terms of dry weight for the PIP in the expressing plant, and these are important in consideration of dietary exposure as well as issues related to resistance management for certain traits. Finally, or not finally, the next step is the biochemical characterization of the expressed traits, which include, again, we're looking at primarily proteins in this discussion, so it would include things like the polyacrylamide gel electrophoresis to show the size of the expressed protein. It could also include Western blot analysis or antibody recognition of those proteins as a means to characterize the expressed protein being what we or what you intended to have expressed in the plant. In addition, the primary amino acid sequence would be expected to be provided. Oftentimes this is deduced from the DNA sequence that was inserted. There's also the expectation to address the possibility of post-translational modification and provide information on the heat stability and digestive enzyme resistance of that protein that's expressed. In regards to the subsequent need for possible testing of the PIP substance itself, oftentimes because of the difficulty of isolating and purifying the PIP protein from the expressed plant itself, there's the use of an alternative expression system for generating test substance in great enough concentration so that it can be used in either the dietary assessment or non-target organism tests that may be required. In the case of the use of alternate expression system for the test substance, the expectation is that there would be the same systems used to characterize that alternate test substance that were used to characterize the plant express system substance in the plant. The final part of product characterization is basically providing a residue analytical method for the inserted PIP trait. If there is dietary exposure for the PIP trait, it's expected that the registrant would provide a method to detect that trait in commerce, and this is basically based on a protein detection methodology and would be expected to be applicable to testing of the raw agricultural commodity. That's the part I have on product characterization. And I think what I'll do at this point is proceed on with the human health assessment category and wait for questions at the end of that section, if it's all right with Chris.
It sure is.
Thank you, Chris. The next slide includes the human health assessment. There are three basic assessment categories, and again, this is geared toward addressing the safety of proteins that may be expressed in the PIP plant. The three categories are in vitro digestibility, and a bioinformatic assessment looking at amino acid similarity. And finally, there's also the consideration of heat stability of the protein that's expressed. In addition to these endpoints, which are used basically to address the allergen potential of the newly expressed protein, there's also the possibility of having to perform an acute oral toxicity test with the expressed protein. The in vitro digestibility assay is basically used to demonstrate that the protein is degraded into small peptides or individual amino acids in solutions containing digestive enzymes such as pepsin for stomach or trypsin for the small intestine. To date, only gastric fluid has been tested for cryoproteins. If you look at the decision documents that have been generated for the registration of these proteins. And this is because these proteins are known to be stable in intestinal fluids, and it was deemed to be not necessary to show that they were not affected by trypsin. Sometimes, however, the proteins are subjected to both simulated gastric and intestinal fluids separately. There's also the effort in the EU at the current time to look at a sequential system for addressing the potential degradation of proteins in the basically simulated human gastrointestinal tract. We haven't seen any studies to date that indicate that this type of testing would be necessary. And we look forward to our EU colleagues providing us with information about the applicability of these tests for risk assessment purposes. Occasionally, when we have seen pepsin degradation data, it's indicated that at the time point that was chosen to indicate degradation, say 1 hour or 2 hours, that the protein was still detectable. In cases where there's an indication that the protein is recalcitrant to pepsin degradation, we've suggested that the registrant look at a kinetic analysis using multiple time points to actually show whether the protein is degraded in a reasonable amount of time that would reflect transit through the stomach or gastrointestinal tract in order to address the possibility that the protein is not rapidly degraded. The next part here is basically looking at a bioinformatic analysis for the amino acid sequence that you have for the protein that's expressed. And that would be using a bioinformatic tool such as the basic local alignment search tool, otherwise known as BLAST, which allows you to basically compare your protein to similar proteins that are in the GenBank or NCBI databases. And that provides information about the nature of the protein that you have expressed in the plant and any similarity it may have to other proteins that have been sequenced. In addition to that general information relating similarity to other proteins, there's specific guidance in the Codex Alimentarius related to identifying the potential for a new protein to be an allergen. And this includes a BLAST search that would look for 35% amino acid identity over 80 amino acid stretches, and it would also include an 8 amino acid match based on the fact that epitopes that are recognized by food allergic people have been shown to be 8 amino acids at the very minimum. And I would point out that there are available special databases for examining the allergens specifically. There's one provided by the Food Allergy and Anaphylaxis Research Program at the University of Nebraska, which is called Allergen Online. And then there are efforts currently underway to generate a similar database called the COMPARE database from the International Life Science Institute. The final part of this particular aspect of the human health assessment is looking at the ability of the PIP protein to withstand heat or the conditions of food processing. And this allows the agency to address the possible different exposure aspects that may be related to the PIP protein in the diet. So if the protein came from a source that was always cooked or processed before consumption, there may be an issue related to the expression of that protein in a plant that would be consumed raw without any heating or processing. And next we have the acute oral toxicity. One of the bases for addressing the toxicity of proteins primarily through the oral route is that when proteins are toxic, they have been shown to be toxic at very low levels. And so when tested in acute oral exposure at high levels, oftentimes the levels that are tested in the acute oral toxicity test is at or approaching the limit dose, which is 2,000 to 5,000 milligrams per kilogram body weight in the rodent species. When there's no expression of toxicity at that level, there's no expectation that the protein would be toxic. However, there are also things that may trigger longer-term testing. When a protein has been shown to have actual adverse effects in an acute oral test, when the protein is related to an anti-nutrient, such as avidin or, say, a trypsin inhibiting protein, these things could actually inhibit the absorption and beneficial uptake of proteins in the diet, and those would need to be examined for possible longer-term toxicity issues. And there's also the possibility, while we've been stressing proteins in this assessment approach, there's also the possibility that non-proteins could be expressed in the PIP-expressing plant. And the expression of non-proteins would require a different approach other than simple oral toxicity, considering, first of all, the previous dietary exposure and any issues that may be related to the non-proteins that were expressed as a PIP. And that concludes, I think, the human health and environmental assessment, or the human health and product characterization portion of the presentation, and I'd be happy to entertain any questions on those two subjects.
Do you have any questions from the attendees? Yeah, I realize it was all crystal clear, of course.
Give me a little more time, Chris. I'm a real slow typer, so.
Well, I can understand that. Yeah, I'm the hunt and peck with two fingers myself, so. All right, I think what we're going to go ahead and do then, not seeing any questions pop up, is we'll go ahead and take a lunch break, and let's say 1:15 p.m. we'll reconvene the webinar. So thank you, and see you then.